In this lecture, I will be talking on cells of the immune system. All body cells, including cells of the immune system, come from stem cells, and stem cells are classified based on their potency or their ability to differentiate into different cell types. For example, early totipotent stem cells appear and disappear within the first few days of gestational age. Then pluripotent stem cells take place in forming different cells of the body. For example, ectoderm cells develop into skin and nervous system. Endoderm cells develop into respiratory organs, liver and pancreas. Mesoderm cells give rise to bone, cartilage, blood vessels, muscle, heart, and kidney. Multipotent stem cells give rise to hematopoietic stem cells. These cells are responsible for formation of different cells of the immune system. And hematopoiesis begins in yolk sac early during the embryonic life, but however, within the third month of gestation, hematopoietic stem cells migrate to liver and to spleen. And within the 7th month of gestation, hematopoiesis takes place within the bone marrow. And as can be seen here, this is the hematopoietic stem cell, which has the capacity to differentiate into hematopoietic stem cell without differentiation and into differentiating hematopoietic stem cell. So this differentiating hematopoietic stem cell differentiates further into myeloid progenitors and lymphoid progenitors. Lymphoid progenitors have the capacity to give neutrophils, eosinophils, basophils, mast cell, myeloid dendritic cell, monocytes, which later differentiate into macrophages, red blood cells, and platelets. Whereas lymphoid progenitors from the hematopoietic stem cell differentiate further into T cell, B cell, natural killer cell, and lymphoid dendritic cell. So now I am going to talk about B cells or B lymphocytes. Actually, B lymphocytes originate from bone marrow and interact first with the stromal cell of the bone marrow. Then they rearrange their membrane-bound immunoglobulin M, IgM, and it becomes immature B cell. Then later rearrange their membrane-bound immunoglobulin D. So B cells with membrane-bound IgM and IgD, they become mature and exit the bone marrow to the secondary lymphoid organs, where they encounter non-self antigens here. And as can be seen here, this is the cortical area. This is the lymph node, the cortical area, which is B cell area. They encounter an antigen here, and then they interact with T cell, as can be seen here, in order to produce antibodies against these antigens. T cells originate from bone marrow and migrate to the thymus gland as precursor, as can be seen here. This is CD4 negative, CD8 negative. Then within the cortex of the thymus gland, they interact with thymic epithelial cells, where they further differentiate into CD4 and CD8 positive, and then further into either CD4 or CD8 positive, during which they arrange, or their pre-alpha, it has two membrane bound or chains, alpha and beta chains. So when they enter here, the alpha chain is immature, so they migrate to thymus gland also for maturation and for development of the repertoire and for the education. And only good T cells will be allowed to exit from the thymus gland, around 5%, where they reach secondary lymphoid organs, for example, this lymph node, and also they reach spleen, and they interact with cells, B lymphocytes, if they encounter an antigen, and it provides B cell with help. Natural killer cells. Natural killer cells, these are actually granular. It is lymphocyte. It is around 10 to 15% of peripheral mononuclear cells. And as can be seen here, they have membrane-bound inhibitory receptor, and also these vesicles contain perforin and granzyme, and they play a major role or a crucial role in killing virally infected cells and cancerous cells by means of perforin or and granzyme in a process known as apoptosis. Eosinophils, these are granulocytes from myeloid progenitor of hematopoietic stem cells, and it produces cytokines and granules, major basic protein, eosinophil cationic protein, and eosinophil derived neurotoxin. These, they have anti-parasitic activity, and also it has a capacity to produce some fibers or filaments from their chromatin, known as extracellular traps. Neutrophils, these are the abundant cell within the circulation, constitute around 40 to 70% of the circulating white cells, and it is multi-looped, as can be seen here, 3 loops or more, and it plays very crucial role during the inflammation. Mast cell, this granular cell, and it is around 10%, and it plays very crucial role in allergy, and also it has an anti-helminthic effect. And as can be seen here, they produce primary mediators, for example, histamine, platelet activating factor, eosinophil chemoattractant factor, neutrophil chemoattractant factor. So they play also a role during inflammation by producing neutrophil chemoattractant factor. And also they produce secondary mediators, for example, leukotrienes and prostaglandins, which they play a role in pathogenesis of bronchial asthma. And also they have the capacity to produce extracellular traps, and these traps actually are very sticky and has the capacity to trap different organisms. Basophils, Basophils are around 0.5 to 1% of the white blood cells with S-shaped nuclei, and basophil they play a role in Helmintic infection. Actually, it has an anti-Helmintic that basophil, as can be seen here, produce interleukin 4, and this interleukin 4 induce M2 macrophages, arginase 1, that kill helminths by depletion of arginine. Also, proteases from basophil recruit monocytes, neutrophils, and eosinophils to the area of infection during inflammation. Monocytes and macrophages, these actually come from bone marrow granulocyte monocyte progenitor, which further differentiate into pro-monocyte, where they leave the bone marrow for the circulation or for the blood, and differentiate into the monocytes. In couple of hours, maybe 8 hours or more, they differentiate. They become bigger, enlarge, with more service molecules, and they differentiate into tissue macrophage, and also it has the capacity to give rise to myeloid dendritic cells. And monocytes and macrophages, they produce cytokines, very important cytokines, for example, tumor necrosis factor alpha, interleukin number 1, interleukin number 6, and service molecules, for example, complement receptor molecule and toll-like receptors molecule. Dendritic cells, they acquire their name from these are the dendrites of the nerve cell, and they have projections which are very similar to dendrites of the nerve cell. So they acquire a name from this similarity. Actually, there are 4 types of dendritic cell. 3 of them come from common myeloid progenitor from hematopoietic stem cell, and these are Langerhans cell, interstitial dendritic cell, monocyte, and myeloid dendritic cell, and lymphoid dendritic cell. It comes from common lymphoid progenitor. So dendritic cell is very important antigen presenting cell, as it is constitutively produces or express major histocompatibility class 2 and B7. So accordingly, they are the most important antigen and presenting cell, and the most professional antigen presenting cell. However, there is a 5th type of dendritic cell. It is known as follicular dendritic cell. It is present within the germinal center or follicles, and it has no phagocytic activity. And follicular dendritic cells, they do not express major histocompatibility, but however, they provide B cell with anti-apoptotic signal at the germinal center. Thank you very much, Professor Ahmed Bola.